Okay, good morning, everyone. Thank you for coming to this distinguished lecture, and we're very honored to have as our featured speaker Professor Tom Poulos, who is a distinguished professor in the Department of Molecular Biology and Biochemistry and Pharmaceutical Sciences, and also in the Department of Chemistry. He wears a lot of hats. He is without a doubt one of the most prolific and impactful scholars, not only in the School of Biological Sciences, but also in the field. I mean, so he certainly belongs in the Biology Hall of Fame. And we're joined by his children in the front row. So we have Jason, Tom, and Nicole all here. So let's give them a round of applause. So structural biologists are not like normal people like the way you and I are, right? They just see the world a little bit differently than the rest of us. So I have to tell you a funny story. So shortly after I became dean, about 10 years ago, Tom contacts me. He says, "Congratulations on becoming dean. By the way, the school logo of the DNA helix is backwards, and you need to fix that." And so I was afraid this morning he was going to ask me what the proper orientation was, so I looked it up. And so I found this, which is really appropriate. And it says, the DNA of every organism on Earth is a right-handed double helix. But it's even more amusing when these photos are used in marketing by life science companies. The most common flaw is to use a left-handed double helix. So thanks to Tom, we had him sign off. We have a correct logo for the School of Biological Sciences, so we could all be proud of that. So Tom has done remarkable work in the field of structural biology and has really profound implications for pharmaceutical development to help treat a number of diseases. And so without any further ado, let's give Tom a round of applause and a welcome.
Well, thank you, Frank, for that introduction and honor of being invited to give this talk. Now, I first want to address what is an obvious question to some of you in your mind, is why I'm sitting in this wheelchair. So let me just talk about that for a couple of minutes. So about 2 years ago, I broke my neck in a ski accident, which resulted in spinal cord injury. And it's very important to get into rehabilitation as quickly as possible. But fortunately, here at UCI, we have outstanding spinal cord injury experts. These folks help direct us towards what is perhaps one of the best rehab hospitals in the country, which is the Craig in Denver, Colorado. So my wife, Andrea Tenner, and I, and she is also a distinguished professor here at UCI, moved to Denver for 3.5 months where I underwent intense physical therapy. And our kids that Frank introduced took time out from their busy lives and careers and came out and helped out Andrea to watch over me during this period of time. And I also want to thank the UCI community generally. I've received a lot of support all the way from the chancellor, the vice chancellor, Dean LaFurla, my 3 chairs. I'm so important, I have to have 3 bosses. Celia Goulding, Andre Luptek, and at the time, Doug Tobias. And their faculty, colleagues, and friends have been very supportive, helping me to get reintegrated into the UCI community. Okay, now let's talk about structural biology. So we're going to have a little primer course here on what structural biology is all about. It's all about determining the 3-dimensional structure of proteins. And proteins are big, complicated molecules. And here's an early example. This is the first protein structure to be solved. It's called myoglobin, and it has an iron in the middle of it. And that iron binds oxygen. And this is how we store oxygen in our muscles. That's the reason red meat is red. All right, and when there's a red meat, it means there's a lot of oxymyoglobin. When it's kind of brownish, there's oxygen there. There's no oxygen there, just myoglobin. And as proteins go, this is actually a pretty simple molecule, but still it's rather complicated. So why do we want to do all this? Well, it turns out about 890 proteins are known drug targets, and about a third of those are enzymes. And enzymes, of course, are the engines of life. They catalyze the reactions. That is, they speed up the reactions that keep us going. And so if we know the structure of these proteins, then we can more effectively design molecules that bind to them and alter their activity. This thing floating in the background is a kinase molecule, and that does a lot of regulatory functions in cells. And sometimes they can go nuts, and cells make too many of them, especially cancer cells. And if you can block the activity of those, then you're going to kill the cancer. And so this is a well-known drug called Gleevec, fits into the active site of the molecule and knocks out its activity. So let's talk a little bit about how proteins are put together. Proteins consist of amino acids, which are small molecules that are linked together by covalent bonds. We have 20 different amino acids, and the order of those amino acids dictate the function of the protein. Now, the average protein in biology has about 300 amino acids. The largest known one has 30,000. But they don't stay as this long piece of spaghetti. They fold up into a distinct three-dimensional structure. And this little computer simulation shows the folding of a 55 amino acid protein. Well, as far as I know, that's the only protein that actually folds up to music. But you get the point. It forms a nice three-dimensional structure. So how do we determine the structure? There's basically three techniques that are used these days to determine structure. The first to come along was crystallography. Then a little bit later, nuclear magnetic resonance spectroscopy. And more recently is cryo-electron microscopy. Now, all three of these techniques have their strengths and weaknesses. But they are complementary to one another, and UCI has expertise and instrumentation in all of these areas. Now, I'm going to focus on crystallography because that's what we do in our lab. So the first thing you have to do is get a crystal. And atoms lined up on a crystal lattice diffract X-rays. What that means is the X-rays interact with the electron clouds around the atoms, and they're scattered in different directions. And that scattering pattern was recorded on an electronic piece of film. And the position and intensity of those spots is related to how the atoms are situated in three-dimensional space. So you take a picture, you rotate the crystal, you take another picture, and you do that hundreds of times. And then you take all that stuff and stuff it into a computational black box, and out the other end comes what's called an electron density map. And that has the electron density distribution around all the atoms. So our job is to take molecular models and get them to fit to this electron density in the correct order. Say our protein has 300 amino acids. So we have to take all 300, fit them into this electron density map. And this is sort of the old-fashioned way we did it with brass models. And you have to put them in the correct order, and then you get the structure. So how long did this take? Okay, my very first protein structure, it took me 11 weeks to collect enough data to get into the black box. The purification and crystallization of the first one wasn't too hard. This whole process took 3 years. Now, it wasn't that I was particularly slow, but that's about what it took in those days. And so back when I got started, there was about one structure solved per year in the entire world. Well, that has all dramatically changed owing to some major technological advances. The black box has gotten a lot easier. This is all push-button software now, so it's easy to use. But one of the main things is the development of what are called synchrotron radiation sources. This is a gigantic storage ring that can have a circumference of close to half a mile, and it's housed in a building that looks like this. And this is the inside picture of what one of those things looks like. And electrons are accelerated around that ring at near relativistic speeds, and the energy given off is right where X-rays are. So we can do a diffraction experiment. And what's the advantage of this thing? Well, the intensity of the X-rays coming out are many orders of magnitude brighter than we can do at home. So what took me 11 weeks can now be done in about 15 minutes. And that, in the black box part, has also gotten a whole lot easier. So if you give me a crystal, we'll get you a structure in probably a few weeks, maybe a month or two at most. And that has resulted in an enormous increase in the number of structures solved. So this is the most recent information from the protein database. If someone wants to publish a structure, an X-ray structure, cryo-EM, or NMR, they've got to deposit it in the protein database. And you can see that has been an exponential increase in all of this. Back when I started, there was only 32. And there are 94,000 unique structures out there, and close to 250,000 that include various complexes, mutants, and so forth. Now, why has this been so important? In this group of proteins are a lot of drug targets. And this is all available to the pharmaceutical world and academics to do with what they want. Okay, and let's talk a little bit about active sites of enzymes, because that's what we're going to target. And I picked a really simple example here, which is an enzyme we need to use glucose as an energy source. The main point I want to make here is that the protein is giant. This thing has 600 amino acids and a little teeny glucose molecule. But it has to go into the active site. Phosphorylation occurs, then it goes away. So when we're going to target things to knock out an enzyme activity, we're going to focus on the active site. So you can think of this as kind of a lock and key problem. Here's the active site. And one particularly good example is penicillin. Bacteria make a particular enzyme that they need to replicate and grow, and penicillin binds to the active site and knocks out its activity. So that's the active site. You can think of this as the lock being the active site, and the key is the drug we're trying to design. If we know the size and shape of the lock, we can more effectively design the proper key to go into that lock and alter its activity. Okay, that's a lot of talk, but does it really work? Right? Well, one of the early spectacular examples was the HIV protease. So HIV, of course, is the causative agent of AIDS, and it requires a protease, an enzyme, to grow and thrive and maintain an infection. Well, this is the crystal structure of the protease, and the surface diagram nicely shows the active site. And so molecules like this were selectively designed to fit into that active site and block its activity and kill the virus. Now, this turned out to really streamline the development of drugs that went to market. So back in those days, you couldn't sequence entire genomes. So each of the genes either had to be separately cloned out. In the case of the protease gene, it was actually synthesized. It was put into a bacteria, protein was made, the crystal structure was solved by 1989, and then the first wave of HIV protease inhibitors came out 6 years later. That's breakneck speed. It normally takes well over a decade to do this sort of thing, and the structure played a key role in doing all of this. Now I want to give you a couple of examples of what's happened here at UCI. The first I'm going to talk about is a collaboration with an industrial company, and the other is nitric oxide and neurodegeneration, melanoma, bad bugs. That may sound like a lot of mumbo jumbo, but I think it'll make sense as we go along. So this first one is a collaboration between our lab here at UCI and Gilead. And Gilead is a biotech company up near San Francisco that specializes in antiviral drugs, especially AIDS. Now, this story starts with one of our favorite enzymes, Cytochrome P450. Now, when we eat nasty stuff, and our body thinks that drugs are nasty stuff, they go into your liver where they get metabolized, get broken down. So our liver is kind of a garbage can, and P450 is the garbage man or garbage woman. So what P450 does is oxidize molecules. It sticks an oxygen atom into a CH bond to give us something that looks like this. And why do we do that? Those kinds of molecules are easier to eliminate from the body. And so it's a clearance mechanism. Now, in biology, throughout the biosphere, there are hundreds of thousands, maybe more, P450s. Almost all organisms have P450s. We humans have 57. And so a rather complicated nomenclature has been developed to tell one P450 from the next. And so it's given the abbreviation CYP. CYP stands for cytochrome P450. And those numbers we don't need to worry about. But this is a diagram showing all the P450s in our liver that are responsible for drug metabolism. And what really stands out here is 3A4. And that's responsible for metabolizing about 30% of all drugs that are taken. Now, I saw a commercial the other day on TV that was advertising a particular drug, and it said, do not take this if you are taking a CYP3A4 inhibitor. So if you see that commercial, you all now know what CYP3A4 does. Now, this naturally brings us to the topic of red wine and grapefruit. And you're saying, what on earth is Poulos talking about? Well, that came out of a study done on the effect of grapefruit juice on cyclosporin. Cyclosporin is an immunosuppressant drug, and it is metabolized by P450s, broken down. And what was found is that patients drinking grapefruit juice needed less cyclosporin. And that's because grapefruit and red wine have this kind of molecule which blocks P450s and prevents the metabolism of cyclosporin. So patients needed less. Now, drug companies know all about this. And so P450 inhibition in this case is considered to be a bad thing. Now, if you go to the drugstore and get a prescription and roll the bottle over, which probably nobody does, and with some of these medicines, it'll say, don't drink grapefruit juice while taking this stuff. Why is that? Because in addition to 3A4 being in your liver, it's also in your gut. So the first thing a drug sees when you take it is 3A4, and it gets broken down. And so drug companies formulate, grapefruit has these kinds of molecules which block 3A4. And so drug companies know about this, so they formulate the dosage knowing that a certain amount of their drug is going to get broken down. And there's a potential for overdose. And this is especially important for opioids. Oxycodone is broken down by 3A4, and if you drink grapefruit, maybe I shouldn't be telling people this, but if you drink grapefruit juice, the oxycodone is going to have more of a kick. All right. So inhibiting 3A4 is not a good thing. Some drug companies, including Gilead, took a different view of this. And this came out of studying this molecule, ritonavir, which is a protease inhibitor. And, in studying its metabolism, they found that it's a really potent 3A4 inhibitor. In fact, it's a better inhibitor of 3A4 than it is of the protease. So Gilead said, well, maybe we could use this molecule as a booster. That is, if we block 3A4, then maybe other drugs will work better. So in the drug cocktails that Gilead sells now, ritonavir is used as a booster. And here's how it works. EVG, I actually can't even pronounce that thing, but let's abbreviate it, EVG. That's an integrase inhibitor. Integrase is an enzyme that AIDS needs, that the HIV virus needs to grow and replicate. And patients need 400 milligrams twice a day for this to be effective. That's a whopping drug load. And the breakdown products are toxic, and that has a lot of bad effects on the patients. However, if you give 100 milligrams of ritonavir, you only need 50 milligrams once a day. And why? Because ritonavir is blocking 3A4 and preventing its metabolism. So this is routinely used now as a booster. Now, structure-based drug design in 3A4, that's what we'd like to do. The problem with this enzyme is it's not a simple lock and key. This enzyme is designed to metabolize a huge number of compounds, and this is just a short list. So the active site is not a simple lock. The lock must be changing shape depending on what the key is. So Gilead came to us and said, can you guys figure this out? So this project was taken up by Dr. Irina Sevryakova in her lab, who's an outstanding P450 structural biologist, and she was able to solve the structure of ritonavir complex to 3A4. And this shows how ritonavir works, or how it binds. That mesh is the electron density cloud, and the yellow thing is the ritonavir molecule sitting in the active site blocking activity. Very importantly, we learned a lot about structural changes that are going on in the active site. And you get a little closer up view of it here. This yellow part of the protein moves the most. So Gilead supported this work for a while, and then Irina leveraged what she had learned from the support from Gilead into NIH support, which she's principal investigator on and has continued to this day. She teamed up with the former student in our lab who got his degree in pharmaceutical sciences, now works at AbbVie, Eric Samuels. He's an outstanding medicinal chemist. So together they designed some novel ritonavir-like molecules, some of which turned out to be even better inhibitors than ritonavir itself. And so Irina solved a huge number of crystal structures, and this is the summary of a lot of work. This is a superimposition of a large number of structures, and what came out of this is what's called a pharmacophore. It's basically a set of rules on what it's going to take to make the ideal inhibitor for 3A4. So we know which regions of the inhibitor must be rigid, which must be flexible, and what kind of properties, whether it's water-loving or whether it likes hydrogen bonds and so forth. And so all this is out there for companies to do with what they want. And so, patent disclosures have been put out and all this stuff. So if anything really comes out of this, Irina ought to get a little kickback, I hope. Now, there's a little interesting side story that goes with ritonavir. Gilead was very interested in converting ritonavir into a molecule that would no longer inhibit the protease. So it would be just a 3A4 inhibitor. That way you might be able to use it more generically. Well, Gilead did have such a molecule, but so did another company. And Gilead felt that that was infringing on their patent. Now, to prove that, they would have to show that it was not easy to figure out how to do this. In order for this to hold up legally, they needed an outside objective consultant. So they hired Professor Poulos as an ACE consultant. And they said, could you figure this out for us? Is it obvious how to do this? And so a team of lawyers was about to show up in my office. And before they came in, I had been mentoring a kid that was about to get into high school, okay? About to get into high school. And he saw what I was doing, and he said, is that a protease inhibitor? I looked at him and said, yeah. I said, how did you know? He said, oh, It's that OH group there. I said, what would you do to get rid of the protease activity? Oh, I take off that OH group. And so granted, this kid was off the chart smart. And he said he would, he read physical chemistry textbooks for fun. But the fact is, any of my sophomore level biochemistry students that were actually paying attention would know that that's what you'd have to do. So I told Gilead that I now had some very unhappy lawyers in my office, and that was the end of my very short consulting career with Gilead. So let's turn to the last topic here, which is nitric oxide and how it relates to neurodegeneration, melanoma, and pathogenic bacteria. So nitric oxide, or we're going to abbreviate it NO, is a highly reactive, colorless, odorless gas whose main claim to fame has been as a minor component in greenhouse gases. And some of my colleagues in inorganic chemistry like this molecule too. But your blood don't flow without NO. It turns out it's an incredibly important signaling molecule in biology. And why is that? Well, our arteries and veins, the inner part where the blood flows, are lined with what are called endothelial cells. And endothelial cells secrete nitric oxide, and it causes vasodilation. So NO controls our blood pressure. Now, prior to this discovery, all that was known is that endothelial cells secrete something, and it was given the name endothelial-derived relaxing factor, or EDRF. Nobody in their right mind thought that a molecule like NO would be so important. And it became Molecule of the Year and was awarded the Nobel Prize for this discovery. Not too long after this was discovered, the enzyme that makes nitric oxide was worked out. And it turns out to be classic P450 chemistry, which is why we got into this. So arginine is a simple amino acid. And what this enzyme does, and we abbreviate it NOS, it hydroxylates, put an OH group on that nitrogen, and it gets blown off as nitric oxide. Now, initially, we knew it was involved in blood pressure regulation. But then it turned out there's another form of the enzyme in our brain that's important for neurotransmission. And there's a third form in our immune system involved in what happened. NO is a free radical. It's highly reactive. So it's used as a defense mechanism for infection. Now, you'd think the first commercial or application of this would be in some therapeutic setting, but that's not the case. The first thing I could find is a hand warming cream. So this stuff is loaded with arginine. You put it on your hands, you absorb it, and it gets converted to nitric oxide. You vasodilate and warms your hands. I have no idea if this stuff works. Another thing out there in the market that you could get at 24-Hour Fitness is some stuff called NO Explode. Same idea. You eat the arginine, gets converted to NO, vasodilate, you oxygenate your muscles, and you get a pump. Well, you get a pump all right, but that's because this stuff contains 2.5 equivalents of caffeine. So that's total nonsense. What does work is beet juice. Beet juice has nitrates, and we have bacteria in our mouth and elsewhere that can reduce nitrates and nitric oxide. So if you want to get an NO boost, drink beet juice. Now, how does this tie into neurodegeneration? Well, a stroke is a good example. In a stroke, a blood vessel breaks, and the region near the break is starved for oxygen. And so the brain undergoes ischemic brain damage, not enough oxygen. But one of the things that was found somewhat unexpectedly is that nitric oxide levels go way up. Now, we need a little bit of nitric oxide in our brain for neurotransmission, but too much is bad news because NO is a reactive free radical. So inhibiting that process would be good for something like stroke. It has since been found that increased levels of nitric oxide is associated with a large number of neurodegenerative diseases. So this is a great project for structure-based drug design. Want to get something into the brain to knock out the enzyme in these neurodegenerative diseases. The problem is there are 3 types of NOS in our body. The one in the brain we abbreviate nNOS, the blood pressure one is eNOS, and the one in the immune system is called iNOS. Now we want to block iNOS, that's the nNOS, that's the target. But we don't want to hit eNOS because it controls blood pressure. So we might be able to knock out the problem in your brain, but if we make you hypertense, you risk heart attack. So that's not what we want to do. So the sequences of the enzyme came out fairly quickly, and our good friend and colleague, Rick Silverman, at Northwestern University took a brute force approach on this. The sequences made it pretty clear that the active site of all these different forms of NOS are going to be very similar to one another. So arginine-like here, you can't get much specificity here. But as you wander away from the active site, there's more sequence diversity. So that's indicated by the red shapes here. These are different amino acids on the surface here, and they're different from the ones in eNOS. That's the idea. So you keep this arginine-like, but you put on variable tails and hope it interrogates the surface of the protein, and maybe it'll bind to something down here better than it binds up here. So this is taking a sledgehammer at the problem, grinding out a lot of molecules. And this is just a short list of stuff that Rick's lab came up with. The top part of this is all the same, right? But the tails differ. And they found that some did bind better to the nNOS form. The lower the number, the tighter the binding. And so in order for this to proceed, however, for structure-based drug design, we need the structure of all 3 isoforms. And we were able to do that, and not surprisingly, they look identical. Only those that really know and love these proteins could tell them one from the other. And if we zoom in the active site, they're very much the same. And one other thing that was discovered from the structures is this thing down here. This is a pterin. That's a molecule that's required for activity. And we'll get back to that a little bit later because it becomes important in the pathogenic bacteria part of this. Okay, this was our first big moment in discovering what can control isoform selectivity. Our job was to figure out structurally why this molecule binds 1,500 times better to the nNOS than to the other isoforms. And this problem was figured out by Mac Flinspeck, a former graduate student in the lab, who has moved on into biotechnology, and Wei-Ying Li, my longtime associate here at UCI. I'll have more to say about Wei-Ying a bit later. So what they did was to solve the crystal structure of that very same inhibitor bound to the 2 different NOSes. This is eNOS and this is nNOS. This is the one that binds better. Now you don't need to be a structural biologist to see that there's a difference between these 2. This one curls and this one's straight. So that has got something to do with the increased selectivity. Now this is a big picture view of the whole thing. So this is the active site, and this is a dimer. So one active site's pointing out toward us, and the other is away from us. If we zoom in on the active site, we see the tail protruding out of the active site pocket. And here is a comparison between the 2. These parts at the top are exactly the same. What's different is over here. The inhibitor has a positive charge there, and it's interacting with a glutamic acid residue, which is negatively charged. And there's an aspartic acid over here, which is also negatively charged. So we have 1 positive, 2 negatives. We go over here, this elongates, so that positive charge cannot directly interact with the glutamic acid. Instead, there's a water molecule here. And this has an asparagine residue, which is neutral. We got 1 negative, 1 positive. So our initial hypothesis was that this situation is electrostatically more stable because we've got two negatives and a positive. Two negative charges close together is not happy, and this is relieving some of that. And that's our hypothesis. How did we test it? Well, we can swap amino acids. We can make this aspartic acid over here and make it asparagine over here. And sure enough, this one lousy amino acid difference was responsible for a large part of the selectivity. This immediately led to a simple design principle. We had a molecular ruler now. We knew that we had to keep this arginine-like. We knew exactly how long to make the linker. But instead of requiring the inhibitor to curl, we're going to freeze it on a rigid scaffold. And then we can play games with what kind of tail out here on the other end. So the very first wave of molecules that were designed looked like this. These two nitrogens are going to take the place of those two nitrogens. We knew exactly how many bonds we ought to have between this and this guy. Remember that this had to curl to put this positive charge in the right position. We're not going to freeze it on this 5-membered ring. And this was the very first set of inhibitors that were designed based on, this kind of molecule also has better drug properties. Aromatic rings are better to use than something that looks like this, and this has fewer rotatable bonds. And that's one of the basic rules in designing a new drug. And this worked right out of the starting gate. So these molecules bound exactly like we wanted them to, and they had some selectivity. This one was pretty good. It was about 1,000-fold more selective for the neuronal enzyme than it was for the endothelial enzyme. So this project actually moved along fairly quickly. And this is what we started with. That's its affinity. The lower that number, the better. It had 1,500-fold selectivity. And after several iterations, we came up with a molecule like this that binds 300-fold more tightly than this guy. But very importantly, it has 4,000-fold more selectivity. So this going around the circle of solving crystal structures, going back to Rick saying we ought to do this, they make the molecule, came up with something that has real good drug-like properties. So this moved on into an animal trial. And let me explain what this is. So the model used here is for cerebral palsy. Now, it's not so much that we're targeting cerebral palsy, but this is a test to see if these compounds can get into the brain and protect it from ischemic brain damage. So this was developed by Siddhartha Tan at Northwestern University. So what you do, you take a pregnant rabbit, you insert a catheter into the aorta. At the end of the catheter is a balloon. When the balloon is inflated, the fetuses are starved for oxygen, and they're born with cerebral palsy symptoms. And so what we're going to do to test this out is to inject the drug before the balloon is inflated to see if it can get in there and protect the newborns from ischemic brain damage. And this video shows how this works. These are what the newborns look like. They're frisky, they climb all over each other to keep warm. And this is one with mild symptoms. This is severe lower limb damage. So scoring healthy from severe is pretty easy. Now we're going to hit them with the drug first. This litter is all normal. Some with mild symptoms. Okay, This is a summary of a lot of data that Sid developed. And bottom line here is in the control, with none of the compounds, you get only 9% normal. Most of them, at least half of them, are going to die, and many with severe symptoms. With the inhibitor, 83% normal. So this stuff is really working. So why aren't we in clinical trials? One step that has to be overcome is this. This is an outrageously difficult synthesis to make this compound in a sufficient amount to move on into clinical trials. And drug companies aren't going to touch this until this gets simpler. So Rick is working on trying to do that. But even that problem, I suppose you could just throw money and manpower at this and make enough stuff to move forward. And actually, Rick has done that. But the real problem is the human blood-brain barrier. The rabbit fetus has not a very well-developed blood-brain barrier, but we humans do, as we should. We don't want nasty stuff getting into our brain. And our molecules are highly charged, and so they have a hard time getting across the blood-brain barrier. So most of the work now is focusing on hardcore medicinal chemistry to try to improve its ability to get across the blood-brain barrier. So that's where that project sits now. But if we can get it in there, we know it's going to work. Now, one surprise that came along that we weren't expecting is the relationship between NOS and melanoma. And this is what a melanoma tumor looks like. It's skin cancer. If this gets out of hand, it's bad news. I think that this is going to be a good example of how basic research and clinical researchers ought to talk to one another. And this developed out of a collaboration with Frank Meiskin's lab, who is the former director of the Child Comprehensive Cancer Center. And for quite some time, together with Greg Weiss, I was director of the Chemical and Structural Biology program in the Cancer Center. And one of our jobs, I see Marion Waterman out there, she made us go get together with clinicians to talk things over, see if we could build bridges. And so I would talk frequently with Frank. And Sun Yang, who used to be in Frank's lab, nicknamed Coco, she's now an associate professor at Chapman University and actually continuing this work. So I would go to their group meetings, and she presented some results that really perked me up. This is tissue culture of melanoma cells. If you add a nitric oxide donor, they take off. And so that lit me up. And so does nitric oxide have something to do with making melanoma grow? And in order to test that, if you want to see if cells are making a particular enzyme, you do what's called a Western blot. It's the same idea behind a COVID test. So you have an antibody to eNOS, an antibody to iNOS, and an antibody to iNOS, eNOS, and nNOS. Well, antibodies to iNOS didn't light up. Antibodies to eNOS didn't light up, but nNOS lit up like a Christmas tree. So normal skin cells don't make any at all, but these cancer cell lines do make nNOS. When I saw this, I said, holy Moses, we're sitting on top of all these inhibitors. So I gave Coco whatever we had in the lab, and she tested some of these out in tissue culture. And that's normalized to 1. This is cell growth. You add these NOS inhibitors, and it knocks them out. And so this was good enough now to move into a mouse model. So this moved into a mouse xenograft. A xenograft is a mouse that's had its immune system knocked out, so you can inject human cancer cells, and the mouse won't reject them. And this worked. So a number of compounds were screened. These two turned out to be the best. That's tumor growth without anything. That's with the inhibitor. Nothing tells a story than looking at the mouse itself. That's the tumor. If we inject the drug 3 days after the tumor is put in, the cancer cells are put in, the tumor doesn't grow. Now, where this project now sits is trying to decrease toxicity. So the mice get kind of sick with this stuff. So for this to move on, that has to be fixed. And Coco is working on that at Chapman with reformulation. There's an interesting side effect that comes along with these things. It's kind of amusing. Some of the mice grow a tuft of blonde hair, so that might remind you of somebody who's about to run for president again. One final quick topic is NOS and pathogenic bacteria. This is something fairly new that came along. And this came out of a paper that was published from Evgeny Nedler's lab at NYU. He found that NO protects Bacillus anthracis. Bacillus anthracis is the causative agent of anthrax. And nitric oxide protects the bug from antibiotic-induced oxidative stress. So antibiotics can induce oxidative stress, basically starving the cells for oxygen that helps to kill them. The mechanism of how that happens is kind of complicated. I can talk about that later if you want. But these kinds of bacteria, bacilli, do make nitric oxide synthase. So when we saw this, we said, oh, well, we should get involved with this project. And so Jeff Holden, a former graduate student in the lab, picked this up. He went on to have a successful career in biotechnology. So he took a bunch of our compounds that we had for the NOS project and screened them against another bacillus. This is B. subtilis, and it's a non-pathogenic strain, so we can actually use it in the lab. And this is the one that really lit up. This is bacterial growth over time, and so the control looks like this. If you add in the inhibitor, it doesn't do much. And we used acroflavin as a skin for skin antibiotic. And add a little bit, nothing happens. But if you add them together, you get a huge decrease in growth. So this stuff is blocking our model bacteria. So this is good enough now to move into our true pathogenic bacteria, and we chose MRSA, methicillin-resistant Staph aureus. And fortunately, we have a collaborator down at UCSD, Victor Nazay, who's an expert in MRSA, and his former postdoc, Federico Beasley, did these experiments against MRSA. And before we did that, Jeff screened a huge number of compounds. And this is an inhibition curve. The lower the bar, the better the inhibitor. And these two guys lit up. And we selected this one because it had the best affinity. And Jeff was able to solve the crystal structure of that. So in these kinds of experiments, we often substitute peroxide for the antibiotic. Peroxide induces just oxidative stress. The antibiotic can do other things. And we wanted to see if it's affecting, working to decrease oxidative stress killing of the bacteria. So in these experiments, peroxide was used. So this is the wild type growth. Notice that this is on a log scale, okay? If you add the inhibitor alone, you get about a 3-fold decrease. If you add the inhibitor with just peroxide, you get a little bit of a decrease, 3-fold. You add them together, you get a 30-fold decrease. And these experiments show over here that if you remove NOS, this is a NOS knockout, it really is susceptible to both the drug and to peroxide. So this stuff is looking pretty good. Now, I want to get back to that one. Early on, I showed a slide that had this thing, pterin cofactor. And this becomes important in this project because in doing crystal structures, Jeff found that some of these inhibitors actually sit in that pocket. And so that was interesting. So we took a closer look at this pocket and compared the bacterial enzyme with the human enzyme. And this is where that cofactor binds. And that pocket is wide open in the bacterial enzyme, but it's really buried in the mammalian enzyme. And so after doing a series of crystal structures, what we found is that some of these inhibitors do well at displacing the pterin in the bacterial enzyme, but not in the mammalian enzyme. And so that's kind of where this project sits. We've identified a druggable target site, and that's what we're going after. Okay, so let me wrap this up. What can we conclude? Well, structural biology is an important tool that can help streamline the development of drugs, but it's very important that we work closely with chemists. This can only work if you've got the chemist and the structural biologist talking to one another frequently. And you can see too that how building bridges between clinical researchers and basic researchers is also quite important. And universities can and should play an important role in early phase drug development, but we don't want to lose track of that our basic mission here is research. So finally, I want to highlight a couple of folks in the lab. Vidi and Jessica both earned their doctoral degrees last fall, and they're continuing along as postdocs in the lab. They're P450 jocks and have kept our publication stream going. Vidi picked up the bacterial NOS project. Christine makes the NOS isoforms used in these studies. I think we're one of the few labs that actually can do that, and hands them off to Wei Ying, who solves the structures. Now, Wei Ying has been the glue that has held the lab together since we got here. He moved with me from Maryland, so he's been here over 30 years, as I have. He's not only held our lab together, but has also, and trained postdocs and students, but has helped out in other labs as well. Irina, you've heard me talk about, she showed up at Irvine shortly after Wei Ying and I arrived, and has been a principal investigator on NIH grants on her own for quite some time. And Alec is a senior researcher who splits time between our lab and that of my good friend and colleague in chemistry, Andy Borovic, and he really stepped up to the plate big time. After I got hurt, basically took over my role as mentor and, to postdocs and students, and is responsible for our continued stream of publications so we can keep up with Frank. Keep your eye on Alec. He's got a bright future in academic science. So finally, I want to thank Frank for the invitation to give this lecture, and thank all of you for your attention.
Thanks, Tom. That was truly a tour de force, and you were a phenomenal lecturer. We have a little gift for you from the school. It's a crystal anteater, and we will have a reception after this, but we have a few moments from questions from the audience about structural biology and drug design. So, Tony. And there's a microphone circulating around.
Wonderful talk, Tom. So a real naive question. I really enjoyed your recapitulation of 2001 there, but I'm wondering how you actually were able to order the steps as that molecule acquired its final stage. I mean, how do you know that's the progression of events that goes through there?
Okay,
when I saw the movie, I didn't read the book, so.
Yeah, I didn't do this. This was done by it's actually interesting. It's a guy that used to be a hedge fund manager, but he really wanted to study protein folding, so he built a computer to do this. So this is a computer simulation. And so none of this is made up. You turn it over to the computer and let all the stuff inside, the forces that cause a protein to fold together. So that's all computer. That's not a cartoon.
Okay, Peter? There's a microphone.
Tom, fabulous talk. I think I might have understood.
There's going to be a test, so.
Yeah. This question is asked on behalf of the EVC and Provost. In the age of machine learning and artificial intelligence, do you really need all that expensive equipment anymore? And
well,
a follow-up to that is, in the old days, it was very difficult to solve the structures of big molecules, and so you'd see a structure of part of it. Has that changed in the field at all?
That's changed quite a bit because of cryo-EM. That could look at very large structures. But if something is disordered, there's nothing you can do about it. But attacking really large things now is possible because of cryo-EM.
I noticed too in your graph of publications solving structures, there was a sharp increase during the pandemic. Is that because people were staying home and actually?
No, the thing, yeah, it's interesting. A lot of the data collection at those synchrotrons is remote. So we stuff our crystals in a dewar with liquid nitrogen and ship them off to the synchrotron. And Stanford was able to keep their synchrotron open through COVID because there was a real push to design drugs based on knowledge of the COVID protein structures. So that moved forward pretty rapidly. So it didn't slow down at all.
Thank you, Tom, for
Hi,
this.
Mahtab.
Hi. Thank you for this amazing, as always, presentation. The question that I have is not a super smart question because I'm not a structural biologist. I can't even.
Oh, you've known me
follow
long
figures.
enough.
I've known you for long enough. So the question I have is how we can improve industry-academia collaborations, because sometimes industry shies away because of all the bureaucracy and all the.
I know.
Yeah.
You're exactly right. I've had a number of interactions with companies, and they roll their eyes back at the policies of the university because the university wants to own all the patents and all that sort of thing. What worked with Gilead, because I knew people in the company, it was basically a gift, an unrestricted gift to do what we want. So, I really don't know what the answer to that is. I mean, on the one hand, I think the university policies are okay because, after all, if it's discovered here, UC should hold patents. So I think that with big companies, I think you could hold the hard rules, but for smaller companies, I think maybe we should change because patents are their lifeblood. And so we ought to do a better job working with smaller companies to help them out. But, we just have to keep pounding the table.
Luckily, Ronnie Hanekak's here, so she can help with that as well. Any other questions before we break for the reception? Do you have another?
Would like the
Oh.
question to be answered about the big equipment.
Yeah.
Peter did ask my question, which is, could you say anything about the role of, the modern algorithms, machine learning, artificial intelligence?
Yeah, there's a thing called AlphaFold out there, which I think they have to deposit their structures in the database. Isn't that right? Yeah, so a lot of AlphaFold proteins are out there. I'll tell you, the problem is, though, if you want to get the overall fold and general idea of what it looks like, that's great. But if you want to do the kinds of things we did with NOS, you've got to have very high resolution, accurate structures. And I'm talking about within angstroms. So until AlphaFold can do that for us, I think there's still a place for all this hardware we've got around here. So, hell, we still need the money to do this stuff. Yeah.
Well, Tom, thank you for breaking down a very complex topic and making it understandable to the masses. We really appreciate that. So let's give him another round of applause for that. And real quick, before you all break, I just want to take a moment to introduce our new Executive Director of Development, Hazel Breen. She's up in the front. As you probably heard me say before in the past, we're kind of like PBS. We depend on viewers like you. And so while we have done very well in terms of our Brilliant Future campaign goals, we have a lot of research programs and needs that need to be funded. So if you're interested, meet Hazel. She should be your new best friend. And so let's all break for the reception. And thanks again, Tom. That was
Thanks.
wonderful.
Right.